I move rapidly to the answer and question and answer slot of time. We are just a couple of minutes late. And I have just received the questions for Jean-Loup. So Jean-Loup, please be ready because we have 3 questions from the audience. First of all, I apologize for the audience because I will pronounce very badly the names. They are difficult to me. The first question is from Seri Kolesnik. And the question is, we are aware of the great progress in the development of non-testing methods and in silico models applicable for single chemicals. Would you be so kind to tell us about any existent, if any, in silico models applicable in any way for toxicity assessment of mixture or automation of grouping of chemicals?
Yes, thank you. Start my camera again. Well, yeah, there are quite a number of tools that are available. There is a mix. There's a mixture calculator developed by DTU. Am I correct, Bruno? And maybe that's a kind of fairly automated tool, right? That is available open access. Is this correct, Bruno? Yeah.
Yes, we have currently the mixture calculator from DTU, which has been developed. Yeah.
So that here's an example where you can actually automate the calculation of risk using. I mean, basically, it's probably using available toxicological data, and then I don't know, your input is exposure, and then you get your resulting risk, right? Correct?
Yes, but still, I mean, they are very basic calculators that are available for the moment. But I think maybe it's worth to mention that, I mean, I didn't mention this in the presentation, but we do have the intention to further develop platforms which will actually facilitate this type of calculation. So we are investigating, for example, partnership with the Dutch authority on the RIVM. And we really want to develop this kind of platforms where you will have open source tools available that can be used for automated calculations of all these risks. Yeah.
Yeah, and then with regards to these new approach methodologies, the NAMs, there are quite a number of QSAR models investigating, for example, the toxicity. The example of these I've given. And there are quite a number of research projects that have generated, like the Euromix tools, which are implemented within the MCRA. For example, looking at adverse outcome pathways for liver and neurodevelopmental toxicity. There are lots and lots of examples. We are also working in EFSA on implementing generic PBPK models that would handle mixtures. So that's further work. And that's being done for human health, animal health, and also for species of ecological relevance. I hope I've answered the question.
I think so. So we can move to the second question that comes from Nertila Kurai. And the question is, besides risk assessment of mixture, another pressing issue is that of risk assessment methodologies for complex chemicals like endocrine disruptors, particularly relevant in the realm of pesticide risk assessment, and nanoscale chemicals, which are generally deemed hazardous regardless of the exposure dose expressed as a function of mass. Are there concrete efforts for elaborating effective risk assessment models for such chemicals?
Yes, so there has been a Horizon 2020 project which was focused on these complex mixtures of compounds which have underground activities. So lots and lots of work in this area. Also, the endocrine screening program of the US EPA has developed a lot of methodologies to actually handle that. I mean, the issue, if we're going to have if there is a classification, it's a bit like, you know, carcinogens. If there is a classification based on hazard only, in order to basically perform your risk assessment, you would still need to have exposure and some kind of threshold values like reference points for your endocrine compounds at some point to get a risk metric. So again, the quantitative aspect need to be further developed maybe in this field.
There is a last question from Marco Hatem Vaqueiro, which is what institution is in charge to finally set the dose that is considered toxic, individual or combined, and what is the criteria to select the publications that meet the minimum requirements to be taken into account in order to make the analysis?
Well, so for example, if we have a mandate, let's say the panel on contaminants, I don't know, let's say cadmium in food, then it's the working group of the content panel will set the safe level, for example, for cadmium. Now that is performed now according to a number of methodologies that we've developed, including the weight of evidence guidance document that we published in 2017, which describes a range of methodologies you can perform. We also have the systematic review document, you know, providing details on how you evaluate the literature, how you would perform a systematic review, and so on and so on. So, I mean, that's a short answer to this question, and I would recommend you know, the person to actually have a look at those documents. But EFSA would set the safe level for some of the compounds, of course, we're evaluating, whether they're regulated or whether they're, but it's also, you know, there are lots of different cases. So I try to answer this question quite concisely, but it would take me a little more to go into details.
Thanks a lot, Jean-Loup. We now move to Armando. Armando, the first question regards any strategy available to perform some kind of detoxification of food or feed. No name of the person that asked the question.
My first answer to this question is that you must be confident that whatever you do, it is allowed by law to do, because there are many ways of chemically or physically destroy mycotoxins, but some of these approaches are not allowed in many countries. So indeed, it will be possible to remove or to degrade the toxins, but on one hand, you must be sure that it is allowed to use that approach. One other issue is that most of the times the regulation states the quality of the raw material that you use. So your raw material must be already in a good shape. So it doesn't matter if you have a process. Let me give an example. If you have patholine in grape juice and you produce cider, during fermentation, patholine is degraded by yeast. So your final fermented product will not have the toxin. However, you must be sure that the apples that you use to produce cider are according to the regulation in terms of the residues of pathogen that are present. So this, I don't know if I have fully answered the question. The best way is always to prevent the accumulation of mycotoxins. So to act as early as possible to avoid fungi from producing and accumulating mycotoxins in crops.
Okay, I move to the next question, Armando. I don't know if it is specifically for you, but it was asked to you. Have antibiotics and mycotoxin and as a secondary metabolites of fungi the same consideration in the food safety assessment?
The difference between a mycotoxin and an antibiotic is very small because some antibiotics are produced by fungi. The main difference is that I may be somehow rude in how I answer this, is that the main difference is that mycotoxins affect humans and antibiotics supposedly will not affect humans, at least using the concentrations that are recommended. Patholine and citrinine, about 40 years ago, were thought to be antibiotics and good alternatives to penicillin, but the toxicity revealed that they were also toxic to humans, and they were classified as mycotoxins and no longer as possible antibiotics. Even so, I believe there are some regulations for the presence of a few antibiotics, maybe penicillin, in foods, because some of these compounds are produced naturally by fungi in crops. So crops may have a small amount of these compounds. So there are some regulation about the presence of some of these antibiotics or precursors of antibiotics in foods.
Okay, thank you, Armando. And now we move to Bruno. And Bruno, the first question is, what is the statistical criteria in terms of sample size and the variability of samples?
I think for me the question is not fully clear, but in terms of sample size, if we mean the number of samples taken, in principle, it all depends on which is the threshold for regulatory considerations established by the risk manager. So if there is the interest, for example, to go for a 95th percentile of your exposure distribution, then you might have sufficient with a number of 60 samples in a certain food item. However, if your risk manager is asking for a very high level of consumer protection, like in the area of pesticides, the level of consumer protection is a 99.9th percentile of the exposure distribution. In that case, you need to move towards around 3,000 samples. So it all depends really on the criteria that are also being established by the risk managers and the question that is asked to us. I hope that clarifies a little bit.
Yes, I think it is. The second question is, in your opinion, the deterministic approach for exposure modeling will be obsolete sometime soon and therefore unapplicable?
No, I don't think it will become obsolete. I think that is one of the messages that I try to give is when a deterministic model can be perfectly valid when you are doing, for example, chronic assessments. Generally speaking, they are quite valid, the deterministic models. Really probabilistic modeling, you need to go towards this type of probabilistic modeling when you need to answer certain questions on uncertainty, for example, or when you want to perform certain simulations, which you cannot do in a deterministic model. But in certain cases, a deterministic model is perfectly fine. And of course, considering how resource intensive these probabilistic models are, like still today when we are running, we are currently running some probabilistic calculations. They run a whole night. You know, it's not something that you can do just with a click of a button. So that's why deterministic models will probably still stick around for quite a while.
Okay, thank you very much. There is a last question from Veronique Siro, and the question deals with the occurrence data. For probabilistic assessment, data on multiple chemicals on a same food sample are needed. But monitoring plans are often independent for the different chemical families. It seems not so easy to get concentration data in a same food sample. How do you manage this limitation?
Well, at least this is indeed something that is a bit difficult when you go across domains, and this is something we will need to investigate further. At least what we have already done in EFSA is we are no longer collecting our data by chemical domain. In the past, we were collecting the data for pesticides, veterinary medicinal products, chemical contaminants, etc., separately. From now on, we are collecting them as one set of data, and that at least allows member states to submit those connected data when they have it. But it's true that in practice still these chemicals are measured in different laboratories. I think this is also something we could consider in collaboration with member states, that although samples are sent to different laboratories, maybe they could get a common identifier before being sent to the laboratories, which would actually allow us to connect the data, even if they have been measured in different laboratories. So this is something we need to start thinking about together with member states, because in the end, they are producing those monitoring data and they are our data providers. Yeah.
There is now a question to all the speakers. I don't know who wants to answer. What attention, if at all, is being devoted to the new paradigm of exposome in the risk assessment models under consideration today? The exposome refers to the lifetime of exposure coming from both inside and outside of the body to which a person comes in contact. Is the concept going to be incorporated in risk assessment models soon? I don't know if any of you wants to answer the question. Maybe Jean-Luc or Bruno.
Well, I think maybe Bruno might be more on the exposure side, but maybe one thing that we can say is that if we're considering the hundreds of thousands of chemicals around, it might take quite a while to be able to assess, all these individual chemicals together. That's.
Well, I think what is Jean-Luc, maybe I think what is important to mention is that exposome is a very theoretical concept, which has a strong validity. I'm not saying this is not a good concept, but the fact is that having the full picture of all the chemicals we are exposed to through all possible pathways, this is never going to happen. I mean, it's not possible. I think what we at least are trying to do, as I said in my presentation, we will look into integrating this non-dietary exposure. And then looking at a lifetime, this is actually something we are already doing with those different consumption data we have for the different population classes, etc. So with good simulations and with good data that allow us to connect internal exposures with external exposures, and this is something Jean-Luc is very familiar with, I think we will get to some concept which is getting close to the exposome, but this will take still quite a few years before we get there.
And one thing I wanted to add to what Bruno just said is that, of course, we are exposed not only to xenobiotics or, chemicals, contaminants, pesticides, and the rest, but we eat kilos and kilos of food every day. So if we're looking at exposome, we need to consider the substances that are in fruit, vegetables, and so on and so on. So that is also the other side of the coin. Huh? It's not just chemical. There are so many nutrients. There are so many, oligoelements, vitamins. So if we're going to investigate to get close to the exposome, we need to consider these natural substances that are in our diet as well. Fibers, and so on and so on. So that's a bit of a job. Huh?
I think the concept is to have an open mind, but as mentioned by both of you, it is not really applicable because we have too many substances to be considered. So there are two further questions, but because we are 3 minutes late and the break is only 10 minutes, I suggest to send the question to Bruno, and then we will find a way to answer also the last two questions. So it is time to have a short, very short break, and we will see again at 11:30. Thanks a lot to the speakers. I have no time to thank you, but I think you did a great job.